Let us turn our attention to the first group of biological molecules we are going to explore in this topic, and that is going to be the carbohydrates. Yum, yum, yum. My favorite type of biological molecule, but it just happens to be because I have a sweet tooth, and you shouldn't be surprised that everywhere I work, I always have something sweet to get me along the hours of grading. Here I have my little gummy worms I'm eating right now that I'm preparing to make this presentation. Okay, so carbohydrates are going to be associated with sweet things. And that is because one of the most common forms of carbohydrates are going to be the monosaccharides. The word mono means one, and saccharide is just another word we use in biology for sugar. So taking the word together, monosaccharide really means one part sugar. Examples of monosaccharides are going to include glucose, fructose, galactose. In fact, I have two of those monosaccharides on my screen. Here you see a representation, linear representation of glucose. Here we have fructose. Later on, you will see glucose arranged in the form of a ring. And that is because in solution, glucose doesn't stay a linear molecule like you see here, but actually arranges in the form of a ring. So monosaccharides are going to be really important to cells as fuel. It's going to be a direct source of energy for cells, for metabolism, for growing, for moving things from one part to another part within the cell. Glucose is going to be the universal fuel molecule in biology. All cells we know can obtain energy from glucose for performing their day-to-day activities. Monosaccharides are also going to be important as raw ingredients for building other polymers, other complex molecules in biology. For example, you know that if you don't burn all the calories you consume, especially glucose, sweets, sugars, your body can begin transforming chemically those sugar molecules, glucoses, and putting them into the form of a fat molecule, which can be stored for later use. The problem is sometimes we don't end up using that later, and it just stays in our body as fat. I'm speaking for myself here. Don't be insulted. Glucose and monosaccharides can also be used as ingredients for making amino acids. And you will see that sugars, monosaccharides, are also going to be part of DNA and RNA. Before I move on, I wanted to show you something. So I have two molecular structures for monosaccharides. Here's glucose, here's fructose. And I wanted to highlight to you the importance of recognizing along the way functional groups we identified in a previous presentation. So here we see, for example, OH, that is hydroxyl. Earlier we learned that hydroxyl is something found in alcohols, but hydroxyl is also found in sugars, usually when there's a presence of another functional group, which is going to be the carbonyl. Carbonyl groups at the end of the carbon skeleton, like you see in glucose, are going to be forming compounds also known as aldehydes. So this aldehyde right here is going to produce a sugar that is not as sweet as the other one you see over here, fructose. The carbonyl group is in between carbons in the carbon skeleton, and this is going to be forming a compound known as a ketone, and ketones have a tendency to taste sweeter in our mouth. The two of them are going to be structural isomers. The only difference is going to be where the carbonyl group is going to be located. But if you count all the hydrogens and oxygens and carbons, the number is going to be exactly the same if you look at one molecule and compare it to the other. So those are the monosaccharides. Cells can take monosaccharides and combine two into the making of a disaccharide. Remember that taking two parts, two monomers, two single building blocks, and connecting them is going to be possible by means of a reaction known as dehydration reaction or dehydration synthesis. So we can take one glucose and the hydrogen at the end of one of these glucose molecules in combination with the hydroxyl group, now we recognize it, I hope, of another monomer, another glucose, these hydrogen and hydroxyl are going to join to make water. This water is going to be going out, it's going to leave as a byproduct. So we are taking water out. That's why this is called dehydration. And there's going to be a synthesis of a larger molecule, that's the joining of the two parts to make a two-part sugar known as a disaccharide. So di means two, saccharide means sugar. Disaccharide literally means two-part sugar. Examples of disaccharides in biology include maltose, which is the example you see here on the screen. Take glucose and connect it to fructose. Glucose plus fructose makes sucrose. Take glucose and connect it to another monosaccharide known as galactose. Glucose plus galactose makes the disaccharide lactose, which is going to be abundant in dairy products like milk. Even butter can have some traces of lactose left in it. Fruits are going to be excellent sources of sucrose. Germinating seeds, nuts, if you like cashews, if you like walnuts, those are going to be types of food which are going to be rich, abundant in maltose. Okay, so now we've seen the monosaccharides, the disaccharides. Can we make more complex sugars? And the answer is yes. Polysaccharides, like the name implied, are going to be many part sugars. And these are going to be long chains of monomers. In biology, this usually happens to be glucose, and these polysaccharides have many uses in cells. Some polysaccharides are going to be serving as storage, and some polysaccharides are going to be helping produce protective structures for cells. Let's take a look at examples. One example of a storage polysaccharide is starch. Plants can store large amounts of glucose, connect them by dehydration reactions, forming one or two of different forms of starch. And I will show you examples of these in just a moment. So when plant cells store excess glucose, they do it in the form of starch. What about us? Can we also store excess amounts of glucose? Yes, we can. And we do it in a different form. It's going to be glycogen. And so after a meal and you have all the sugar in your blood that hasn't been taken up by cells in your body, the liver can take all that sugar and go through the process of dehydration reactions, combining the sugars into a polysaccharide known as glycogen. Later, as you go through your activities, between one meal and the next meal, which may come hours later, those glycogen molecules begin to be broken down little by little by hydrolysis so that your cells can have a steady supply of glucose and not fatigue because they ran out of energy. Another example of a polysaccharide is cellulose. But cellulose is going to be an example of a structural or protective type of a polysaccharide. Cellulose is a compound we find in abundance in the cell walls of plants. It is what makes a cell plant rigid. I'm sorry, a plant cell. It's what makes a plant cell rigid. It's what allows wood, for example, to be wood, to be something hard and sturdy. And so this cellulose is going to be also made of many glucose molecules attached to one another. And one more structural polysaccharide we can find in living things is going to be chitin. Chitin also helps make cell walls in fungi, but it can also be seen in layers on the outside of the body of the animal, forming part of the integument. The integument is like the outer covering, the cuticle for the animal. In insects, in crustaceans like lobsters, crabs, they are going to have this structural polysaccharide known as chitin. Let's take a look at the images. Now I'm going to see if I can I'm trying to magnify. the image I have on my screen. So here you can see starch. And I told you before, glucose sometimes is going to appear like a ring, like a hexagon, and that is what happens in solution. So starch, notice how it's going to be forming like springs, like coils, and that is what allows glucose molecules to be packaged neatly inside of structures in a cell known as plastids. So amylose is one of the forms of starch we can find in plants. Another one is going to be amylopectin, and this is going to be a little bit branched. Notice that amylose doesn't have the branches, but amylopectin does. Here we have glycogen, and this is how we are going to be storing glucose. And notice how heavily branched this is, but it's still going to be glucose attached to glucose, attached to glucose, many glucose molecules connected by dehydration reactions to make this storage type of polymer. And then down here we have cellulose, and you can see that in cellulose, the glucose molecules are going to be forming straight chains. And that is because we have two kinds of glucose. We have alpha glucose and beta glucose. Both of those are going to be found in cellulose. And it is the form, an alpha glucose connects to a beta glucose that allows for this linear arrangement. And this is convenient because with these long cables, bundling them together by means of hydrogen bonds, which you can recognize by these dotted lines, a plant can make bundles of these cellulose molecules arranged into a microfibril, which is going to be the matrix that helps form a cell wall in a plant cell. And I mentioned that in animals and also in fungi, we can find another type of a structural polysaccharide, and that is going to be chitin. And if you look at the foundation, it's still going to be a modified glucose. Here we have glucosamine, There's going to be an amino group that is going to contain nitrogen in the amine, and there's also going to be more of the carbon that is going to be extending off to the one side. This group of atoms, which has the amine, the nitrogen, CH3, and there's a carbonyl group over here too, that is going to be known as the N-acyl group. So all the glucose molecules in chitin are going to be attached to one of these N-acyl groups. When you think, for example, about how hard the exoskeleton of a crab, of a lobster can be, it is going to be because of chitin. A fungus, like the mushroom part of a fungus, can grow straight up, and that is going to be because of the support obtained by something like chitin. And so that's the carbohydrates, monosaccharides, disaccharides, and polysaccharides.